Ebola, Listeria, C. difficile. Bad news of germs and viruses travel fast. Make no mistake, though, not all microbes are bad. In fact, some can even improve your health. That's according to our guest, Jason Tetro. He is a microbiology researcher as well as the author of the Germ Files. Hello.
Hello.
Okay, let's get into it. There's your book. There it is up on the screen. Your book is subtitled. Let's get into the subtitle. The Surprising Ways Microbes Can Improve Your Health and Life, and How to Protect Yourself from the Bad Ones.
Exactly.
So let's cut through the swath. What separates the good microbes from the bad ones?
Well, first off, when we talk about bad microbes, we're really talking about pathogens. And they're only about 0.1% of all the microbial species that are out there. We always give them most of the media attention, but in reality, the other 99.9% fit into one of three categories. One is commensal or inert. They just, eat, digest, leave. Another one is symbiont, which means that these bacteria and viruses actually will work with our bodies to improve our health. And then there are antagonists. They're called pathobionts. And when they get a little bit too high, they kind of, annoy our bodies and make us feel not so well. So, of course, the goal is to associate more with the commensals and the symbionts and not so much with the pathobionts and get rid of the pathogens.
Okay. I'm still calling them good and bad because I'm not saying any of those fancy science words because I'm going to mispronounce them. Okay, so most of them are good. We tend to hear about the bad.
Well, of course we only hear about the bad because at the end of the day, if you are thinking about what is important to Canadians and Ontarians, they want to find out how not to get sick, right? So that's why we really do focus on the bad ones. And most of the time when I'm talking in the media, I'm always talking about some virus, Zika or some pathogen like Ebola. But the fact is we have so many good germs that are out there that really are not getting as much attention. It's changing because now we're starting to hear more about how microbes can improve our health. And as a result of that, we're starting to see a little bit of a shift. It's only a small shift at this point, but I'm hoping that with this book, people can understand that, the whole collection of microbes can actually improve our lives on a day-to-day basis.
All right, we'll get into the good ones in a moment, but sometimes good things turn bad, bad things turn good, and you talk about this in your book. I'm going to read just a short passage from it. Here's what you wrote. Figuring out why a friend becomes a foe is a real challenge. When a bacterium turns on us, it's usually because of something we did, probably inadvertently, or something that occurred in our bodily systems, probably in order to keep us alive. It all comes down to stress. Bacteria hate a stressful environment, and when they encounter one, they choose one of three paths to deal with it: fight, flight, or sporulate.
Yes.
Okay. What do you mean fight, flight, or sporulate?
Well, let's start with the sporulate first.
Good, I don't want to have to say that word again. It's really difficult for me.
It's okay. Some bacteria have the ability to create what we call spores. And all that means is that they've tightened themselves up into a very little ball that is very resistant. Now, C. difficile is one of them. Very difficult to kill, and they spread throughout the environment. And then when they finally get back to a nice, happy, area, they'll grow up again. Flight is pretty much what it sounds like. Get into the water flow and get out of the system. Now, fight, on the other hand, is something that's extremely important. And one particular example of that is pneumonia. We have bacteria throughout our body, trillions, tens of trillions. And they're in our sinus cavities. Now, they're happy there, okay? But let's just say all of a sudden you get a cold or the flu. Now, your sinuses, as you might expect, are not going to be very happy. And they start to create a number of immunological proteins that are out to kill. And some of these bacteria go, this is not really good. So they try flight. But unfortunately, sometimes they don't come out the nose or the mouth, they go into the lungs. And it's worse there. And so now all of a sudden these... You're
sort of trapped in your lungs.
You're trapped in the lungs, and you've got more immunological proteins, you've got more problems, and you're cornered. So if you're an animal that's cornered, what are you going to do? You fight back. And that's what these bacteria do, and we call that pneumonia.
Huh. Poor little guys fighting back, trapped in my lungs.
Well, that's the thing, is that we always think about pneumonia as being something that is, trying to kill us. Well, no, it's really just bacteria that have been unfortunately put in a situation that is not very good for them, and they're going to try and fight
You're
back.
trying to make us have sympathy for bacteria.
I'm trying to make you love the microbes.
Okay, we'll see if we get there by the end of our conversation. Okay, how do you, so if we have this, how do we lessen the risk, minimize the chances of having problems with bacteria in, I don't know, in your, well, what's it, microbiome?
Oh, the microbiome.
Yeah, what is that?
Yeah, it's a jargon.
Yeah,
And
well, it's a science word.
It's a science word that really fits well in the academic world. All it means is it's the entire collection of microbial species within a given environment. So there's the human microbiome. There's the indoor microbiome. You know, it's a wonderful word to use in academia, but at the end of the day, for us as humans, we really just want to think about two things: our bodies and the environment we live in. And just to give you an example, if you are in a room with no ventilation, over time, the actual number of bacteria is going to rise, as you might expect. But most of the time, they'll be human-based, and because the pathobionts, the ones that antagonize us, can probably live a little bit better in these environments, those numbers are going to rise, while the good bacteria, the symbionts and commensals, are going to drop. How do you change that? Open the window, because now you get fresh air and more good commensals coming in. Or you put plants. The plants are actually going to help increase the number of these commensals.
So diversity is a good thing?
Diversity is everything.
Oh, diversity is everything, not just a good thing.
When it comes to microbes, as it does when it comes to Prime Minister Justin Trudeau and E.O. Wilson, diversity is absolutely everything.
Because it's 2015, although now it's 2016, right? We need diversity. Okay, so what you're saying is that we open the windows, get diversity. Bacteria loves diversity just like Canadians love diversity.
Exactly.
All right. If you don't have a diversity in germs of you, if you're sort of, you were just saying that's riskier for you.
Oh, it is. Because what ends up happening is that if you don't get the symbionts and the commensals in there, then the pathobionts are going to grow. And as I said, if they grow to high enough levels, they're going to antagonize you. So pathobionts are responsible not for an infection. You're not going to get a gastrointestinal disease or a respiratory disease from them. But what you could become is obese, or suffer from diabetes, or cardiovascular disease, or possibly depression. Now, when you think about it from that perspective, you go, okay, I need to drop these numbers. How do I do that? It's actually very simple. You change your lifestyle so that you are doing good for the good germs. And it's something as simple as adopting a diet that's high in fiber, low in saturated fats, low in simple sugars, high in fiber, also known as the Mediterranean diet.
Okay, I've opened the window, I've changed my diet. I'm presuming the next thing you're going to tell me is go exercise some more and all these other things.
Yeah, exercise actually is wonderful. There's these little cells that we make called neutrophils. They make the pathobionts really unhappy and they flight, whereas the germs that we really like, they're like, hey, this is good. You know, we're gonna party away here. And so that ends up helping as well. So really what all of this does is it's showing you that the more that we can improve this ratio, if you will, of the good to the bad, as you like to call it, we're going to improve our lives. Your mouth, right? You don't want bad breath? Get rid of the pathobionts that are creating the volatile sulfuric compounds. Instead, brush your teeth, chew some gum, sugarless, right? Get some xylitol in your body, and lo and behold, your teeth are going to improve. There's even now, believe it or not, there's actually a bacterium that is so good, it is being sold as a way of combating plaque. It's called Streptococcus salivarius.
Is that true? Or is this just being marketed to me?
No, this is actually true. If you go back and you take a look, it's been around for like ever. And now it looks like this is something that's going to be able to be improving children's lives to help prevent caries, right? Like these, and probiotics are the same way for your gut. So you get those lactic acid bacteria, the same ones that we use for fermentation of sourdough, sauerkraut, pretty much anything that's called sour, miso. Those bacteria, you concentrate them high enough, right? Then you take them, whether it be through a pill or through a drink that's actually fermented, and you're going to improve your life. Like that's really what it comes down to. And it's because you're improving the diversity by getting rid of the pathobionts or lessening the number that are there and increasing the number of the good germs.
I live and work in the world of broadcasting. I have a favorite broadcaster? Steve Bacon, since I'm sitting in his chair right now, is the answer to that question.
He's my favorite too.
He is.
Yes.
Listen, you live in the world of germs. So do you got a favorite germ?
Yes, Lactobacillus acidophilus.
Of course it is.
When I give presentations, I actually have a large version of it from my friends at.
Say it slowly again.
Lactobacillus acidophilus.
Okay, what is that?
It's a probiotic bacterium that has 3 things going for it. One, it's like a mafia don. When it gets into your body, it seeks out pathogens and pathobionts and will either kill it by itself or send signals to other good germs to say, we don't like him. Done. And it's amazing. It works. We've seen this in the lab, and you can actually see this in living animals. The second thing it does is it interacts with our bodies. We've actually become used to having this bacterium in us so that it helps to keep us calm. And that is really an important thing. And then finally, the third one is that it actually promotes your body to digest well. And that's so important because once that happens, then you're getting all the good nutrients, you're getting your minerals, your vitamins, all of that. You're getting rid of bile, which is really important for lowering cholesterol. And you're just essentially creating a much happier environment. And just for the record, with enough Lactobacillus acidophilus in your body, what comes out of you probably is a little bit solid as opposed to what some people suffer from.
All right, well, let's just go there then, if we will, because I think some people who are watching us are probably germaphobes, and when you start saying things like that, they get a little repulsed.
Well, yeah, I can understand that. And it's because for, over 100 years, we've been looking at germs as our enemies. I remember this big headline back in 1912, the war on germs, right? Well, unfortunately, every time we've had the war on, insert name here, we've lost or we're losing, right? And the thing is that in the war on germs, we have been losing. And we're going to continue losing until we realize that it's no longer about having a war, but learning to love and appreciate, respect, and have a commitment to the good they do for us. It's what I call the arc of love.
It wasn't so long ago, a number of years ago, that all that dishwashing detergent started to be antibacterial. And everyone started using it. And then we were told, don't use it because you're killing all the good stuff too. And the same thing happened with, which we all used during SARS in Ontario, and antibacterial hand wash. Some people still use these things, others choose not to. If I constantly or overuse this kind of stuff, am I also killing the good bacteria? Where are we on this now in 2016?
Okay. Well, first off, the one thing I have to say right off the bat, because I had this discussion in the back before we sat down, is alcohol hand sanitizers and antibacterial soap are completely different things.
All right.
Antibacterial soap has a chemical in it called triclosan. Now, it was originally thought to become an antibiotic. It just didn't do so well. So then it was sold as an over-the-counter disinfectant, an antiseptic. Don't use it. It doesn't work. The FDA now has basically said it doesn't do anything. And back in 2012, the Canadian government was actually thinking of calling it a toxin to the environment. So if you see triclosan, keep it on the shelf. Hand sanitizers are made with alcohol. 62 to 70% is the most effective. You put it on your skin for 15 seconds, and it lowers the burden, if you will, or the number of microbes that are there, bacteria, viruses, etc. Okay? Yes, if you're doing this on a constant basis, you probably are going to be removing some of the good germs. But I like to say, if you want to get those germs back, show them and yourself an act of love by essentially hugging yourself. Because the minute that you touch your skin in another location, those good germs are going to come back on.
I want to kind of flip this on its head. I'm a parent of young kids, so, we got bacteria everywhere, good, bad, the ugly, everywhere, right?
I have a friend, she calls kids the best Petri plates out there.
Yes, all right. So parents always say, let your kids play in the dirt. We don't let them, but we pretend like we're going to let them. Like, go get dirt. It's good for you. Is it good for you?
Oh, yeah. Okay. I've always been the type that says, let them eat dirt instead of cake. Mind you, not in a dog park, just so you know.
All right, be careful. Yes.
The fact is that, when you are getting out into an environment that is fresh, much like opening the windows, right? Then you're exposing yourself to these commensals and these symbionts. They're coming from the soil. And so by getting into the mud and getting into the dirt, what you're doing is you're actually improving the diversity inside of yourself. So that's really what it comes down to, is you're giving kids the chance to have a better diversity as they're growing up. Now, they really shouldn't be out doing this until they're probably about 7 or 8 months of age. And the reason for that is the immune system is still very it's not mature yet, right? It's still growing. And mother's milk is really the best thing for them in those first 6 months. But then as their immune system starts to expand and develop and become more, become stronger, if you will, then you can start introducing these other microbes. Because what happens is that certain bacteria, which could be pathobionts, can eventually become commensals or symbionts because we can get our immune system to tolerate them.
Let me read something else from your book. At the molecular level, an antibiotic recognizes and destroys molecules associated with a particular bacterial pathogen. In the lab, it's a simple process of adding the chemical and watching the bug die. But changing the landscape from the petri plate to the human body adds complexity and, unfortunately, risks. How does it come to be?
Okay, so first thing is first. When we are talking about any type of drug, whether it be an antibiotic or a pharmaceutical, and we test it in the lab, it's basically an agar plate with some bug on it, and then you add the chemical and you see what happens.
Controlled environment.
It's a very controlled environment. Now add that into a system where you have tens of trillions of microbes. And suddenly
you lose control.
You lose control. So what you have to do is you have to get an understanding as to what might be going on. And over the years, we've essentially understood that when you are using certain types of chemicals, and antibiotics are a perfect example of that, you kill diversity. And it's not just antibiotics. We're seeing this now with proton pump inhibitors, right? The purple pills for indigestion.
Okay.
And when you start killing diversity, as I said earlier, you're giving the chance for those pathobionts to grow, and possibly pathogens. And this is one of the things that we see in a very well-known example called Clostridium difficile. Now, everybody knows about C. diff, right? What nobody knows is, why does C. diff happen?
No one knows that. We haven't figured that out.
Oh, we do. I do.
Okay,
and
tell us.
So what happens is C. difficile is a member of a population in your gut. And it's happy, right? But it's not doing anything. It just sits there. And about 15% of us actually have the C. difficile in our bodies. You take an antibiotic. Now, the C. difficile is going to be resistant to an antibiotic. The other microbes, not so much. So they get wiped out. Now the C. difficile is in a barren wasteland where it can grow. Now, what happens is it's a very greedy monster. So as it grows, it reaches a certain state where it realizes it wants more food. But there's really only one place where it can get more food, and that's you. So it starts releasing toxins. Now, when those toxins are released, it antagonizes you, your immune system, and your pain receptors, really. So now you're all of a sudden suffering from diarrhea, from the immune system trying to flush it out. You're suffering from pain as a result of the pain receptors being hit. And you're also essentially losing the lining of your gut because it's being dissolved and used as food. And then as that happens, you end up with pseudomembranous colitis, which is the big word that they talk about. And eventually, this thing could get into your bloodstream and kill you. And all because you killed the diversity in the first step.
All right, but I mean, arguably, I'm arguing against the germs, they had to give someone these antibiotics to treat something else. So this is sort of a side byproduct of this.
That's a side effect, if you will. Yeah, adverse event, as we like to say.
We always hear we're running out of suitable antibiotics, and we don't have any more, in the pipeline. Are researchers at least trying to come up with I don't know what we call it, a drug of last resort?
We have some drugs of last resort. The thing is the problem or limiting factor, if you will, isn't the drugs themselves. We can make drugs all the time. It's getting them to market, which can cost, I think it's now up to $2 billion. Now, would you spend $2 billion to be able to get maybe 6 to 8 months of effective use before you start seeing resistance? Oh,
meaning it will be the last resort only for a short period of time.
Exactly.
So my investment isn't worth it.
Exactly. And the thing is that we still don't have very many pan-resistant, which are resistant to all antibiotics. We need to be focusing on better use of antibiotics by better diagnostics, better understanding of the microbes that are inside of us, and better understanding of the side effects or adverse events that are going to occur.
And hugging ourselves and spreading our good germs.
We must always hug ourselves.
Don't be afraid of the germs, spread the germs to yourself. That's what your advice is. We're out of time, and on that note, we got to go. Thank you.
Thank you so much.
Help TVO create a better world through the power of learning. Visit supporttvo.org and make a tax-deductible donation today.